Okay, so I think we can make a start anyway. So good morning, afternoon, evening, everyone, wherever you are, and welcome to the Palaeoverse lecture series. This lecture series is focused on software development and methodological approaches in paleontology, with the aim of promoting the use of new tools in the field and to give researchers a platform to showcase their work. With this series, we hope to shake up the usual type of lecture by having a mixed format with time dedicated to the presentation, demonstration, and discussion of the software or method presented. Now, typically these sessions will run for about 50 minutes in total, and we'll run a series throughout the year, generally on the last Thursday of each month, unless they overlap with the holidays. So you can see the slide for these upcoming talks we have. If you're interested in presenting a new method or software you've been developing or implementing, please feel free to reach out, as we're always happy to hear from the community. But before moving on, just a few quick introductions. My name is Lewis Jones, and with me today I have Cecily Nicole, Erin Dillon, and Bethany Allen. And the four of us are on the core team of Palaeoverse, an organization aiming to bring the paleobiological community together to share resources, reach agreed standards, and improve reproducibility in paleobiological research. And if you want to know more about Palaeoverse, please do go ahead and check out our website, palaeoverse.org, and our GitHub organization. I believe those links are being dropped into the chat now. But that's all from me today. Cecily will now give you some helpful tips and instructions for this webinar, and Erin will introduce this month's speaker. So thank you very much, and please enjoy the seminar. Over to you, Cecily.
Thanks, Lewis. So as Lewis just mentioned, the format of the seminar will consist of a talk by our speaker, followed by about 10 to 15 minutes at the end dedicated to the Q&A and discussion. My fellow hosts and I will help to field your questions and moderate this discussion. Please note that the seminar will be recorded and will be made available on Palaeoverse YouTube channel. However, we'll stop the recording before the Q&A period in case you prefer not to be recorded. Before we get started, I'd like to quickly point out a couple of useful Zoom controls at the bottom of your screen. You should see a Q&A button. You can use this to submit your questions to our speaker during the Q&A session. We'll either read out your questions or invite you to turn on your microphone if you'd like to ask the speaker directly. There's also the raise a hand button. You can raise your hand, we'll call on you and invite you to turn on your microphone so you can ask your question. Then we have our chat. So currently you can only chat with hosts in case of technical difficulties, but we'll allow you to chat with other attendees during the Q&A period. Closed captions, you can turn that on and view the transcript at any time. Lastly, I'd like to highlight a couple of ground rules. So our aim as Palaeoverse is to cultivate an open, respectful, and inclusive environment during this lecture series where we can learn from each other. With this in mind, we ask that you please be respectful and kind when engaging with the speaker and the fellow attendees, particularly when asking questions and interacting with others in the chat. Any form of harassment or inappropriate behavior will be grounds for removal from the webinar platform. For more information, please refer to our Palaeoverse Code of Conduct, which can be found on our website. I think Bethany will put a link into the chat now. And with that, I'll pass over to Erin to introduce today's speaker.
Awesome. So thanks, Lewis and Cecily, for getting us all up to speed on how the lecture series works. Today, I have the great honor of introducing our speaker, Dr. Brooke Longfox, who is a data curation scientist at Phoenix Bioinformatics and a research scientist at the South Dakota School of Mines and Technology. So Brooke works at the intersection of ecology, evolution, and data science, focusing on how scientific data are generated, structured, and reused. Her research is interdisciplinary and spans fields such as conservation paleobiology, aquatic ecology, environmental science, and data curation. And through this work, Brooke emphasizes fair and open science and strives to improve data practices across disciplines. She also advocates for equity and inclusion in STEM through the Graduate Women in Science Community and SORTI, the Society for Open, Reliable, and Transparent Ecology and Evolutionary Biology. So we're very delighted to have Brooke with us today with a talk titled Building Open Reproducible Paleontology, a practical introduction to fair data and MorphoBank. So please join me in welcoming Dr. Longfox to our virtual stage. Take it away.
And thank you for the introduction, and I'm excited to be here. So today I'll be sharing more about MorphoBank, which is a data repository for phylogenetic matrices. Okay. Okay, so a bit about myself, like Erin mentioned. So I am a data curation scientist at MorphoBank. MorphoBank is a specialized data repository, and it is currently managed by Phoenix Bioinformatics. MorphoBank is about 25 years old, It was originally run at Stony Brook University by Maureen O'Leary, and about 5 years ago, it was passed on to this nonprofit to manage. So today, what I'm going to do is go over a bit about why morphology matters, why it's important to store this data and to share it with others, a little bit about the reproducibility gap that we're facing right now in paleontology, how fair data helps address this gap, and how MorphoBank enables reproducible research. Then I'll go on to a MorphoBank virtual tour. It's an online repository, and so it's open access. You can access it to access data or to deposit your data. I'll go through an exemplar project and some of the new features because we just had a huge update in October of 2025, and we're still working on rolling out features, but a lot of cool stuff has been added. Okay, there we go. So a bit about morphology. So morphology is essential for studying life in the past, as most of you should know, and it remains central to paleontological inference alongside other methods. So some things, we may have ancient DNA or geochemical signatures or other signatures for studying fossils, but morphology is really what is mainly used to study the fossil record. So morphological character data is used to bridge extant and extinct taxa and phylogenetic analyses. So a lot of phylogenetic analyses these days are using molecular data. And if we try to do anything with fossils or bring this into the past, we really need morphological character data. And so I mention this because a lot of the molecular data is stored in GenBank or other repositories for molecular data, but we have MorphoBank built just specifically for morphological data. These character matrices underpin tree inference and are used for deep time and for any poorly preserved taxa. So here's a little bit about what the matrices look like and how we store them in MorphoBank. So a little bit of how this data works for a tree. So we would have a list of different taxa or species. This is a really simplified version from Berkeley's Understanding Evolution website, and I like to use this just to give a little bit of a background. And so we have these different species over here on the left, and the columns would then be the different characters. So we have different characters like, are there four limbs? Yes or no. These are the different states for the character. Is there an amniotic egg? Is there hair? Yes or no. So these are things like presence or absence, zeros or ones is what they would be coded in matrix. And so this example here for primates, rodents, rabbits, crocodiles, dinosaurs, birds, there's amniotic eggs, yes. And so this is a simplified version of a tree where you can see the relationship. And what this actually looks like in matrix form. So here's another example. This is an example from Tree Thinking. It's a book on phylogenetics, and this is a great book as a resource if you're wanting to learn more about this. But basically, for example, if we have the character tail, and so I'm going to go here down, character number 6 is tail. So over in this matrix over here, we have character 6. So the tail is either elongated or short. So elongated would be a 0, short would be 1. And so for every single taxon listed over here, it would either be 0 for elongated or short as 1. And I'm showing this because in MorphoBank, what we have the capability of is for you to store your matrix that looks like this. And we have the different taxa listed. You can add specimens. You can even link them to specific specimens in museums. And then we have the capability to have the character information and the character states. Okay, so a bit about this. MorphoBank has been around for 25 years, but we have been working on increasingly getting data put into this. We want data into this repository so we can have reproducible research. Reproducibility is the foundation of science, and paleontology in the past has not really been known to share the data. And so you may have a phylogenetic analysis and maybe a supplementary document or a table in the paper that's published that has this data. But really, we need to make it reusable so other people can do the same study or build on the data. I'm going to go over a little bit about this. Repeatability would be if in your lab you're using your data and you continue to use the same data and it's the same setup and the same outcome. For reproducibility, it would be you take your data and you give your data to someone else, and someone else can use that data and come up with the same result. And so this is one application to make sure that we have reproducible and transparent research. And then we have replication. And so this is the reuse of data. So we can, have someone take your data, build upon it, have a whole new study, and come up with some different findings or the same findings. And that's replication. And so some common issues that we face in paleontology is that matrices are frequently unpublished or unavailable after peer review. The supporting images for how the character states were defined are scattered across supplementary files, or sometimes they're just not even there. We have characters that are poorly documented, and so it makes scoring decisions impossible to evaluate. So if you're looking at a previous published project or previous research, and you're trying to decide, oh, well, this specific species was scored a certain way, if there's no pictures or characters aren't documented well, you can't reproduce that, and you're not really sure how to score that. And then data are routinely lost after publication when authors change institutions. So usually once you publish your paper, you are like, okay, I'm done. And you have it out there, you think it's good. But if someone else asks you, hey, down the road, they may email you, please send me your data. You don't always have that, especially if you're moving around institutions or you've just moved on to a new project. So really, the way to solve this is by putting it into a repository. So why do we need reproducible data? You should not have to reconstruct data that already exists. And so your data should outlive your paper and be useful beyond it. There's a lot of different things that morphological data can be used for. In addition to phylogenetic analyses, they can also be used in conservation paleobiology, macroevolution, comparative morphology, so various different fields and subfields and other projects besides just phylogenetic trees. Now, why would you want to benefit, or why would you want to share your data? What benefits do you personally get? So one thing is you get more citations. So if you publish your data usually has a DOI, and you would cite your data in your paper, but other people could also take your data and cite it. And then you're getting citations not only for your research paper, but for your research products such as data. You get more visibility, so your work becomes discoverable and reusable. It also makes for transparent research. One of the first steps you do when you try to publish your paper is it gets sent out for peer review. And peer reviewers are increasingly expected to look at the data and access the data and see if it makes sense with the study results. Then we want reusability. So you don't want your data lost after publication. You want it to be able to be reused or shared with others. And then there's collaboration opportunities. So you want to be able to build your work, and other people may take your data and contact you about it and say, hey, I'm using your data. Would you like to collaborate with me? And we can work on this new study. And then there's also credit. So you've worked hard to create this data set, and so you should get credit for it. So the data on MorphoBank and most places when you deposit your data in a repository, you get a DOI. Specifically, it would be great if that data is linked to your ORCID account. So MorphoBank has a new feature where now when you add your data, and if you have ORCID linked, then that shows up on your ORCID as a data set. Now, besides like personal benefits, there's open science benefits. So some of them are there's no paywalls, anyone can view and use data. regardless of institution access. This really supports equitable science, where researchers from underfunded institutions and countries can fully participate. And then this also has the opportunity to benefit education and research. Teachers and students can explore real data in the classrooms. Okay, so why do repositories matter? Emailing a file to a colleague is not data sharing. Repositories make data findable, usable, and permanent. Some of the things that repositories do are preservation. So data are stored long-term, independent of the author career changes or any institutional moves. The data also has associated metadata. So metadata is the structured context that makes the data interpretable by someone who wasn't in the room when it was created. Then there's searchability. So data sets can be discovered and reused by researchers who didn't even know that they existed. And citations. So DOIs give data sets formal credit. So they link them permanently to authors via things like ORCID. And then there's the peer community. So repositories connect your data to a broader scientific infrastructure already used and trusted. There's a couple different kinds of data set, or a couple different kinds of repositories. There's general repositories. And these are things like Dryads, Nodo, Figshare. These allow you to quickly deposit your data sets, but they're not discipline specific, and they don't really have any kind of curator expertise. So these are essentially just file storage mechanisms. Then there's specialized repositories. These are repositories that need structured data. For example, this is MorphoBank, and MorphoBank is for morphological phylogenetic matrices. Some of you may have heard of MorphoSource. That would be for like 3D scans. So these repositories, they take more time to add your data to them. It's not just a quick drop files in, but it makes sure that the data is interoperable by making it a standardized format. It makes it reusable by maintaining relevant metadata and structure and data organization. And the data is usually peer reviewed by subject matter experts. Okay, so what is MorphoBank? So MorphoBank is a specialized repository that allows you to store various things related to morphology and phylogenetic matrices. One, it can store character matrices. It can also store images. And with images, we take 2D and 3D images, but it does not, you don't even need to actually have a phylogenetic matrix. You can just create a new MorphoBank project and store a couple hundred images of different specimens if you want. And so it's a great way to just focus on any of the data related to morphology. And then there's MorphoBank stores metadata. We have various forms of projects and folios. So folios would be a form of projects for images. And then all of the data that you add to MorphoBank is given a DOI. And so this is great for linking and for citation purposes. Everything in MorphoBank is FAIR. And so FAIR is a specific kind of data framework, and it is fair, or it is findable, accessible, interoperable, and reusable. So how does MorphoBank make data fair? It makes data fair by being findable. So everything in MorphoBank is given a DOI. So that means that the data can be located and cited. It is accessible. So data and metadata are retrievable via standardized open protocols. The metadata is persistent even if the data is no longer available. It is also interoperable. So the exported data format is a standard format, either Nexus or TNT, and you can choose when you download data. And this is the most commonly used files for phylogenetic software. And then the data is reusable. So when you go in and you add your data, you have to specify the licensing. Usually we prefer CC BY, and so that way anyone can legally build on them. So what kind of data is in MorphoBank? The data represents over 1,000 matrices, and really there's a global user community and hundreds of journals represented. We have thousands of scientists from around the world and from thousands of institutions. Here's some just metrics of what is in MorphoBank. To this day, I actually updated these this morning, and we have a way to access online to see what what all is in MorphoBank. All of this data is open access. So you can just go on to morphobank.org, look around at the repository and search for anything, and you can download the data and reuse it. So I mentioned MorphoBank is 25 years old this year, and we have been working on significant improvements to MorphoBank. So in the fall, this past fall, we had a completely new software infrastructure and media storage upgrade, essentially. So all of the code that MorphoBank was built on was updated. And so it used to run very slow. Now it's a lot faster. We have also expanded the phylogenetic tree building. So before we had the option where after you added your data, you could then run a quick analysis in Palp. And now we also have the options for Mr. Bayes and for TNT. We have a new ORCID integration. And so you can log in via ORCID. And what we're working on, I don't think it's available yet, but it will be either this week or next week, is a link to the ORCID that pushes the projects that you published. So if you add data to MorphoBank, it will link to your ORCID and that will be listed on your ORCID account as a data set. We have a new feature. It's an AI character name and state extraction procedure. And so this is not out yet, but it should be out next week, where before you would have to go in and when you added your data, you would have to either make sure your file was in the right format. So we usually had Nexus or TNT files specifically formatted to have character name information. But we found that a lot of people had this information as a separate file. And so now we have a background AI assisted curation process that where you can add multiple files and the AI program goes through and it pulls the information and puts it together for us. And then you can also now, besides Nexus and TNT files, you can also upload CSV and Excel files. We have a new capability to combine different OTUs in the matrix. So before in the matrix, if you were looking, you could separate just specific characters and look at those. Now you can combine things. So you could put like multiple species under one genus and like kind of collapse them. And so we have variations of working with these matrices now updated. And we have a new user interface, which is much quicker, and user experience improvements. Now, a bit about funding. So MorphoBank is free to use. Most repositories are free to use, but they're not free to run. And so, like a natural history collection, the specimens are open to all, but curating and maintaining them still requires sustained investment. And so, what we ask for everyone, MorphoBank in the past has been fully funded and run by NSF. NSF funding helped do all of these version updates. But now we are looking at a community membership model where we ask the community to contribute to funding and sustaining the platform. So if you are working on submitting a grant proposal, and you have to have a data management plan, and you want to use MorphoBank as your repository, it would be great if you could add a little line about MorphoBank and say, I'll donate or provide so much funding to support the platform. We also have two different membership options. One is an institutional membership. So this is universities, research institutions, museums, societies that are interested in supporting data preservation. And we're always looking for new members. And so if you think your institution is interested, please reach out. We also have the option for individual donations. And so this is if you like using MorphoBank and you just want to support it, we're happy to take individual donations. This is a list of everyone who currently supports MorphoBank. We have various societies, museums, universities, and this is places all around the world. And so thank you to all of these different groups that support this repository and make data reusable and fair. And if you need to contact me sorry, my cat's climbing around back there. Before before I show a tour of MorphoBank, if you need to contact me, it's BLONGFOX, so blongfox@morphobank.org. You can also email our general email, which is curator@morphobank.org. And so now I'm going to switch to looking at MorphoBank's website. Oh. Okay, so if you go to morphobank.org, this is the home page. I'm going to make it a little bit bigger. Okay. And so every time you refresh it, there'll be a different set of pictures here, and these are all pictures from different projects. And you can scroll around and kind of see a bit of metrics about MorphoBank. You can search for different things, different projects here. And we have some featured things. And then we have a thank you to our supporting members. First, I'm going to go ahead and look at an exemplar project. It's 773. Okay. Okay, so this is what a published project looks like. And this project is essentially the data set. The project is this one discusses what paper the data was published in. So this is the paper, and it was published in Science. So it has the journal, it has the volume, it has the page numbers for the published paper, and it has the different authors. This abstract here is the abstract from that paper. And then it's a specific image that you can pick whatever you want to showcase as part of the project. And this is what a published project looks like. And if you scroll down, you can click read the article, and that will link to the article. So this is the published research that this data set came from. And then, so there's the project or the article DOI, and then there's the project DOI. So this is the specific data set DOI and what you would use if you're sharing the data with other people. We have summary boxes here. So it shows what the project contains. So this one has close to 13,000 images. There's one matrix for all of these images. There are over 6,000 taxa, over 2,000 specimens, and over 4,000 characters. And then there's attached additional documents that are in this project. We have specific information about the matrix and about the characters. We have the capability where for each person who contributes to this data set, if they log in and create a MorphoBank, if they register and create a profile, then they're listed here. And so this allows us to see who contributed what to the data set. So this, for example, this is the first author, Maureen. She added 81 taxa, 347 specimens, and so on for media notes, characters, and different kinds of cell scoring notes and scoring labels. But we have all of the different people and authors who contributed to this data set on here. And it's going to keep scrolling. So a lot of people added to this. There's information, a taxonomic overview for the matrix. And then this is really cool. So project views, We have the total number of project views. So how many times individuals have opened this specific project page and looked at this project. So this one has been viewed over close to 200,000 times. Um, we have list of, information about media views, who has looked at different things. And so we have some metrics of after it's published, how much or how much use or views this this project has. We also have information on project downloads. So, media was downloaded over 2,000 times. The matrix itself was downloaded almost 2,000 times. The entire project was downloaded 3 times. These are great metrics to provide your funding agency. So if the funding agency supported the research and you're writing a report, you can say, we added the data to MorphoBank, and here are the numbers of how many other people have viewed it or how many other people have downloaded the information. And so that's always great to have. And we also have a summary of that up here on the right. So it has when the project was created, when the project was published, and then a little summary of a couple of the different project downloads and views. Because MorphoBank was originally established and supported by NSF, we have the option where if your research was funded by the U.S. National Science Foundation, there's a little mark you can click and it adds this logo to the project. And then we have a list of the author institutions. So who were the institutions that the authors are affiliated with, and they get added here. So we have the matrix. So this project overview is the main homepage. There's a matrix tab. And so this is the link to the actual matrix. And basically it has all of the details here with how much scoring is there, the taxa, the characters, different information about images. The matrix itself has a DOI. So you could refer to this project, and this project only has one matrix, but you could have a project that has 15 matrices. And so maybe you are reusing a matrix in the project. You would cite the main project DOI, but you would also say the specific matrix I used was this DOI. And then there's the option for download. So you can download a Nexus file without notes, just a plain Nexus file. We have basically a bunch of different options for how you want to download the matrix. You can also just download the character list, so with or without notes. I'm going to go ahead and click View Matrix. This one's a big project, so it's taken some time to load. Okay, so this is what the matrix looks like when you first open MorphoBank. So the first thing it does up here, you can see there's show names plus images. So that's why we're seeing all these images. Now, a typical matrix you're probably used to seeing is, let's see, just numbers. There we go. Is just numbers. And so we have, the taxa are over here in the rows and the columns are the character names. And these cells are filled with the character states. So there's some different viewing options you can do here to look at your matrix. And in terms of characters, so we have the character list. For example, this one is the skull, and the 2 states are absent and present, and there's even notes. And so the notes are great here because a lot of times when you build these characters and character states, you're actually building them from previous research. And so here, this is a citation that this character was from another paper, and some details about it. And really with the notes, you can write whatever you want. So this one, for example, this is modified from another study. And you can even add, this one doesn't have notes, but you can even add a note and say in new character. All of these, you can attach different media. So you can scroll, I'm scrolling through here showing the different media options. So these were all attached to the specific characters. We even have a function, I don't think this one has it, but there's an option you can label things. And so you can say that there's an absence or a presence of something. You could point to I don't know, like something with on the image and put a label there to help understand what you actually mean by present or absent. Okay. So that is what the matrix looks like. This project has a lot of media. You can see 100 per page. This one has thousands of media, but I'm just going to scroll through and you can see all the different image attachments. And like I said, with MorphoBank, you don't even need to have a matrix. If you have a project and you're publishing a paper and you have a bunch of images and you don't want to just drop them to another repository, you could put them into MorphoBank and publish the project as just images. And what's really cool about the images, is they can be attached to specimens. So the first step would be adding the different taxa. And so you can, here's some more that are genus and species. And so you can add the different taxa in, and then you can go into the specimen tab and add vouchered or unvouchered. So for a specific, let me jump here, like to V. Okay. So for this specific species, there's two different specimens that they used as part of this project. They were both in AMNH, so American Museum of Natural History. They were in the Mammalogy collection. And here are the two different specimen codes. And you can see one of them has one image and the other one had four. And so I clicked on that. This is probably slow to load. There we go. But there's four images for one of the specimens and one for the other. We have options. You can add view for media. So a lot of different media formatting. And like I said, there is the option to add 3D media. And then documents. And so you can add whatever doesn't fit as media or matrices, you can add as documents. Say you're doing a combined molecular morphological matrix. MorphoBank will not accept it. You would need the morphology separate, but you could go in and add the combined matrix here, or you could add just the molecular data here or any kind of supplemental information. This, for example, has permissions for copyrights. It has information related to the matrix, any appendices from the actual journal article. So all of that can be added to documents. And documents takes various file types. Okay, so that was an overview of a project. What I want to do quickly is show you how to add your own data. So for example, I found this paper that we did not have in MorphoBank, and this paper does have a phylogenetic matrix in it. And so if I scroll down, and I think I downloaded this, yes. So this, as part of that paper, the data set was in supporting information, and it was a supplemental document, and it just has the information as a Word file. And the same, it was a separate file as part of that paper that also has the character information. And so these two things as Word files make it hard to reuse the data. And so I'm going to go ahead and show you how to create a new project using this as an example. So if you have so you can access the data and download anything without logging in. If you want to add data to MorphoBank, you do need to create an account. So I've created my account. This is my profile. And the first thing that comes up is this screen right here. And then it shows you your unpublished projects first, followed by your published projects. This right here, I'm going to create a new project. Let's sign in. Okay, now create a new project. Okay, so there's a couple things that are required, like a project name. Was it supported by NSF? I already checked this one was not supported for the project name. We recommend you just use the title of your paper. And there is a allow reviewer to log in. So if you if you're still working on this project, you haven't published the research yet, but you're about to send it off to peer review and you don't want to make the data publicly available yet, you can click this and enable reviewer access. And basically you give a password and we have information about what link to give the reviewers and in your cover letter to the editors, you would put to access my data, here is how you get to it, and here is the password. So that way they can share it with reviewers and it wouldn't be public information yet. So we have the option when you're setting this up, is it in prep or review? Is it in press or is it published? So published would only be like if you already have a DOI for the article. Journal title. Okay, so what you can do if you don't want to add in information individually for something you already have published, you can just take the DOI and there we go. We have a retrieve option and now everything has been filled in for me. So it has the journal information, it has the authors, the title. What it doesn't have is the abstract. So I'm going to quickly paste that in because everything that's required here, you need to fill in to be able to move on to actually create a project. But once you fill in this page and you hit save, I now have a new project. And we'll have this soon where right now I'm just adding someone else's data that's not on here. But if this was your data and you have ORCID linked, then this will export to ORCID saying that you have a project. And so here's a bit what the background looks like. Once you create a project, we don't have any information in here yet. But I'm going to go over to matrix and I'm going to create a matrix using this data. So I had to convert this to a Nexus file because it was not. Right now, our new skill we can our new version can take Excel, CSV, and Nexus and TNT files. This is a Word document, so it won't accept that. So I did turn it into a Nexus format, and so I'm going to go ahead and upload that. Okay, and there was no, because so basically I turned this file into a Nexus format. There was no character information, but it was in a separate file. So this is our new AI extractor, and I'm going to add the character information in. Right now, it says we take Word documents, but we're still working on some testing. It wasn't accepting it, so I just did a PDF. And I'll hit extract characters. And I think this takes a little bit of time. Let me see. Oh, I don't have the other option. Okay. So this will take some time to process. But once it's in there, actually, let me see if I can show what. Oh, you know what? I don't have it in here. Okay. So this will probably take a couple minutes. So maybe if we do if anyone has any questions right now, would be a great time.
Hi, Brooke.
I
I think there's a question in the Q&A. Let me just read it. It's a bit long. Um, okay. Okan, do you want to ask your question live, or do you want us to read it out for you?
I don't know if you can hear me. Sorry.
Yep, we can hear you.
Okay. I first of all, before I ask the question, I just want to say thank you all for making paleontology and research more accessible. This is so awesome. It's definitely helped me when I was doing a research project at my uni. But for MorphoBank, what I wanted to ask was, are there any standardized character coding practices to, ensure that data sets match each other and they're consistent and reliable to, address the subjectivity in character conceptualization and coding?
So we try to keep everything in a Nexus file format. And if you're if you're looking for an easy way to build your data set, you can build it on MorphoBank and it will update everything in that format. Mesquite is also used that has that format as well. But really the minimum we try to meet is to make sure that you have, in addition to the matrix with the scoring, to have the character information. So that's the most common thing we see missing is there's no character state names or state information. So it will have the species listed or the taxa listed, but there's like a 0 or 1 next to it, and it says character number 4, but there's no like it doesn't say, like what is character number 4, what does 0 or 1 mean. And it's a search for, let me look through the paper, okay, let me look through the supplementary information. Oh, okay, it was uploaded to this like external file and that's not there anymore. And so in terms of data practices, our goal is just that you have the character information with it. Okay, so this finally uploaded, and now you can see that the character name information and the states have been added to the file that we originally had that didn't have that information. And so now if you go onto anything on MorphoBank and you download it, that information will be there, and it makes it so you can reuse the data or modify it. Say you're working on something and you only want to use, I think, these first couple characters, it looks like your skull. Um, you only want to use the skulls. It's a way to reuse the data without having to like recreate it yourself. Does that
Thank
kind
you
of
so
answer
much.
your okay?
Yes. I was also wondering about like when different I don't know how to explain this. When different researchers put different states as different characters, but uh, you just explained that, so thank you.
Yeah. And I think so. I don't think there's some papers out there that explain how to build good characters. There's not that much out that's new on this. One thing that I've noticed as I curate things, and I can see it right here in this matrix, is present and absence. So usually absent would be 0 and present would be 1, but researchers go back and forth. And even in this example, there's there's the swap here where absent is 0 for one case, but then it's 1 in another case. And so when you're building when you're building your data set, think about these things and try to at least make it consistent would be my recommendation. And try to put notes somewhere because people do reuse these. These are this kind of work is reused a lot in characters, especially if you usually don't make up a character on your own. I mean, you may, but you'll modify it from someone else's work. And so when you're working on your research to have details of this information or how you came up with these states, that would be great.
Thank you so much.
So, sorry, Brooke, did you want
Yeah.
to continue now
Yeah,
or?
yeah. I can,
Okay.
and it's I don't have that much more, and I know we're like working on time. Oh, this one's a very long one. I think that's
So
why it took a
we'll leave
while
the
to.
questions till you finish then.
Yeah, okay. So I've got this in here now with all the characters. I'm gonna click next. We have a a system that goes through and see if there's any like major errors in the matrix. And so it will tell you that. What I'd suggest on that previous page, if this was your own data, you need to go through and like make sure that there weren't any issues. Um, so we use AI now, and it is getting better and has gotten better over time. But when we first started doing this, so this has been in development for 2 years. When we first started doing this, every single matrix that we added had an error where it was changing the wording a little bit, or maybe like forgetting punctuation or swapping character information. And so as part of a to be consistent and to make sure we have good data out there, review everything, just in general, review everything AI does, but review everything in here as well to make sure it's accurate to your work. And so I'm showing this what I would do if I were to publish this, because part of my work is I curate things. And so before I would actually publish this data, I would go back to the original PDFs, the original research. I'd read through it and make sure everything matches up, because I don't want to publish something and have someone reuse it and it's not true to the original research. And so now it's just uploading the matrix. That's taking some time. And this feature right now that I'm showing you is in our beta version, but this is, I think it's going to go out tomorrow afternoon. We're going to release this. And so this will be on the normal morphobank.org. But now you can see the matrix. And I'll switch it to show numbers. This one has a lot of question marks. But yeah. You can also go in and like I'm going to show right here, if I double click, you can edit characters or add characters. And you can add character notes. Okay, so that was just a quick overview of how to create your own project. We take, like I said, we do take media. We take 2D, 3D, CT. You can batch upload information. So if you have thousands of images, that's the best way to do that. We have a bibliography option where you can, so say you build your matrix on other people's work, you can create the bibliography here, add those references, and then you can add any kind of documentation needed. Okay, so that was my quick overview. I know we're like running short on time.